Okay, guys, part four. These are rather lengthy. There's really isn't a good place to chop them up, so I'm just going to fly through some of this stuff. So this part, we can classify neurons based on their structure and their function. Multipolar has three or more processes. Bipolar, two process, axon and dendrite. Unipolar, single short process. Functional, afferent, efferent, and association neurons. Association neurons are interneurons. They shuttle signals through CNS pathways. So you'll have some neurons that communicate, for example, between the two halves of the brain, the two hemispheres. And so those are interneurons. And here is, you can pause this and take a look at this if you so choose. Want to keep going. So neurons are irritable and excitable, and action potentials are carried along the length of the axon, always the same regardless of the stimulus. So an action potential is an action potential. As I always say in class, if you stub your toe or cut off your toe, the action potential is going to be exactly the same. So one of the things that we're going to have to talk about is then, how does the nervous system interpret the difference between stubbing your toe and cutting off your toe, right? That's a difference in the strength of the stimulus. So how do we know some pain versus a significant amount of pain? So that's one of the things that we are going to talk about. Now, I mentioned irritability versus excitability. Irritability, you should already know, the ability to respond to stimuli. Excitability is the process of generating an action potential. So these are only nervous tissue and muscle tissue because these are the only ones that have the necessary channel proteins that allow them to propagate an action potential. Some basic definitions. I'm not going to test you on this. This is just something we're really going to be talking about, potential difference. So if here's the membrane, voltage measured between 2 points, we're going to measure from the inside to the outside, and then we're going to have a resting membrane potential, and then we're going to talk about how that changes through an action potential. This should be a review. So there's a potential on either side of the membrane when there's different number of ions, and the membrane provides a resistance to flow. We've already talked about this, right? There's more sodium outside and more potassium inside. And this membrane is going to block the flow. So that's how we end up with this potential. So the resting membrane potential is really going to be produced by three things: the concentration gradients of ions inside and outside, basically sodium and potassium. The plasma membrane resists flow, and we have the sodium potassium pump to maintain those ionic concentrations. Right, so the key ions are going to be potassium, sodium, and chloride. I don't think I'm really going to talk about chloride. I think my focus is going to be on these two, and I'll leave the chloride for a greater in-depth view of graded potentials and inhibitory postsynaptic potentials and those sorts of things. All right. So the two types of signals that we have: graded potentials and action potentials. Remember what we said, that with the graded potentials, these were going to be from dendrites, or our receptive regions. Action potentials do not change. If I have an axon that is 100 miles long, the action potential here at mile 1 is going to be exactly the same here. Graded potentials diminish over length. And that's important if we're talking about a receptive region. So with these membrane potentials, we're going to have three events: depolarization, the inside of the membrane becomes less negative; repolarization, it returns to the resting membrane potential; hyperpolarization, it becomes more negative. And we can see that here. These are just stimuli. So this is a depolarizing stimulus. Here's our resting potential. Right? So the inside is more negative. Depolarization, it's bringing it to zero, making it less negative. Hyperpolarizing is bringing it further away from zero, making it more negative. All right, so these graded potentials, your textbook will talk about the mechanism behind it. We're not. I just want you to know what they are. They are short-lived and localized changes in the membrane potential, and they decrease in intensity with distance. That's a big one, and this is a big one. The magnitude varies directly with the strength of the stimulus. So a sufficiently strong graded potential can initiate action potentials. Right? So these receptive regions, we only want to fire an action potential if the stimulus is strong enough. So if we have this very weak stimulus, by the time it reaches the soma, it's going to be so weak that it cannot generate an action potential, because the voltage changes in the graded potentials are decremental, meaning they keep getting lower the further away you get from the point of stimulus. And so they can only travel over short distances, and that's why we see them in dendrites, because they are pretty short. And we see that here. So here is our initial stimulus, and the further we go away from that stimulus, we go right back to that resting membrane potential. So we need something pretty strong that by the time it reaches the soma, it will kickstart an action potential, and then the neuron is basically responding to that stimulus. All right, after this, we're going to be talking about action potentials. I'll probably say it in the beginning of the next one. The action potential that we're going to cover is exactly the same as what we did in the muscle, except I'm adding layers. We're going to quantify. We're going to use a graph. It's going to be a blast, but it is exactly the same. All right, that's the end of this part.